Our next session will provide an overview of low-grade gliomas. Our chair, Dr. Claus, will review current knowledge of low-grade gliomas and how engagement with patients living with low-grade gliomas is key to advanced treatment. Please welcome Dr. Claus.
Thank you and welcome again. So I'm going to talk a little bit about the generalities of low-grade glioma. I do want to update everyone on some of the definitions and terms that have come up with the new information from the World Health Organization. Talk a little bit about one of my favorite topics, epidemiology, and what we know or don't know about risk factors for glioma, and then talk a little bit about some of the new research that we are undertaking to hopefully advance what we know about low-grade glioma. So I don't have any disclosures. I do receive support from the National Cancer Institute, as well as in the past from the American Brain Tumor Association. So many of the slides and data that I present here today, I do want to give acknowledgement to the Central Brain Tumor Registry of the United States, which is headed by Carol Krutchko. And essentially, this provides data for researchers. It collects across the country from a wide variety of state registries, as well as other resources, including the National Cancer Institute, data on both benign and malignant brain tumors. So the little pie chart that I have here indicates the overall distribution of primary brain tumors, at least within the United States. And as you probably know, the majority are a higher grade glioma or glioblastoma, but a large proportion are still the lower grade gliomas, the oligodendrogliomas, the astrocytomas. And we estimate that there are about 3,000 new lower grade gliomas in the United States every year. The mean age is about 41 years, although many patients are in their 20s and 30s. And we do see a little bit higher rates in males and individuals who identify as white. But we will say that some of the racial and ethnic reporting probably varies quite a bit by group. So we're not sure if the data is complete for some of the non-white individuals. So what has changed over the past few years in terms of classification of glioma? You might have heard of an organization called the WHO or World Health Organization. And so periodically, not only for brain tumors, but for a variety of tumors, they update definitions based on new information. And so a lot of what has changed over the past few years is the advent of genomic or genetic information. So previously, gliomas were graded as 1 through 4, and essentially based on histology and clinical criteria. So what they looked like under the microscope and how people tended to do clinically was how diagnoses were defined. So grade 1 tumors are usually seen in children. That will not be our focus today. We'll look at tumors that are primarily seen in adults. And so those are the 2, 3, and 4. Obviously, there's always a little bit of overlap between what we consider the pediatric and the adult tumors. So grade 2 and 3. And so just to define, people use low and lower, high and higher in a variety of ways. So low grade or lower grade tumors are what we're going to be focusing on today. And so we're including both the 2 and the 3. So in the past, as I mentioned, these tumors have been defined by histology. So when you or the pathologist looks under a microscope, what do you see? What do the cells look like? What is their nature? How many are there? And so the names at that time were generally called astrocytoma, oligoastrocytoma, which was kind of a combination of 2 types of cells and therefore was also known as mixed glioma, or oligodendroglioma. And I just show a little bit of a picture here. The majority of tumors, at least that we're seeing and that are reported to these registries in the low-grade glioma category, are generally oligodendroglioma. And this is what they typically tend to look like. And you've probably heard this term. It's called a fried egg, in the sense that the center of the cell looks very dark, and then there's kind of this halo that looks like a fried egg. The grade 4 tumors are generally called the high grade, also known as glioblastoma, although some other tumors are now being reclassified to be defined as high grade. And people have used the term de novo if the tumor arises out of the blue, or secondary if it transforms from a lower grade tumor. So this is the article that gives all the details of the new 2021 WHO classification. It is available free, so if you do want to take a look at it, this is the link to do so. So general changes, without getting into too many gory details, is then and now. So as I mentioned previously, it was histology and tissue-based analyses, and the laboratory tests that were done were something called immunohistochemistry. But what is happening now is we're much more able to use information about genes or biomarkers. And so some of the pieces of information that now we know to be of import are mutations in genes that are called IDH1 and 2, and then something that is called 1p19q codeletion. So these are portions of chromosomes numbered 1 and numbered 19 that essentially are missing or switching pieces. And when we see that in the tumor, we denote that as a specific type of tumor, in particular oligodendroglioma. Grading previously used Arabic numerals, this 1 through 4, and the grading was across tumor types. And what happens now is Roman numerals 1 through 4 are used, and the grading is within tumor types. So those are kind of the big high-level changes that we're seeing. So what does this mean for individuals that are diagnosed or have been in the past diagnosed with lower grade glioma? It can mean that a diagnosis that you were assigned in the past may not necessarily be the diagnosis that you are assigned with now. So as we said, gliomas are split primarily into adult and pediatric using this new coding scheme. The adult diffuse type glioma, now there are only 3 categories, and I've listed them in the table that I took from that paper. So the tumor types are astrocytoma that has this mutation, oligodendroglioma that again has this mutation and this chromosomal change, and then glioblastoma. IDH wild type means that that tumor does not have the mutation. And then within those tumor types, grading 2, 3, 4, 2, 3, and 4. So that does mean that some tumor types, and those of you that have been diagnosed, and we're seeing this in our study, some of the tumor types that may have been on your pathology report in the past are no longer recognized. So in particular, oligoastrocytoma, also known as mixed, is no longer a recognized entity. Anaplastic astrocytoma and diffuse astrocytoma, IDH wild type, that was graded are 2 or 3. And additional information that you might find helpful if you want additional details is listed at this link below. Again, this paper is freely available. So this is just a little bit of an update of some of the nomenclature of how things on the left might have been classified given the prior WHO classifications, and how they are on the right now classified in 2021. And so some of the big changes are that the astrocytoma wild type tumors are now listed under glioblastoma. So what practically does this mean? It does mean that persons initially diagnosed with lower grade glioma may need to have their tumors reclassified. And if there is a time period at which you or your healthcare providers are considering additional treatment, a change in treatment plan, we do believe it's important to have your materials re-reviewed and updated to help guide treatment. So moving a little bit to tumor risk factors, it's something that, patients ask us, well, I was exposed to this, when I was a child, this happened. Do you think that caused my glioma? And I will say that the most common thing that people ask me is telling me that a sibling hit them over the head with some instrument of destruction as a child, and did that cause their lesion? And we don't believe that head trauma has anything to do with this. We do now know that genetics, those that are inherited, as shown in family history, meaning that one or more individuals in your family may have also had a brain tumor that might be glioma or might be something else, that there is some increase in risk associated with that. And I'll talk a little bit more about that in just a second. We do know that ionizing radiation is a risk factor, not only for glioma, but pretty much for a wide variety of cancers, other brain tumors, lymphoma, leukemia, breast cancer. But we think that in general, this is associated with high dose levels of exposure. So for example, individuals that were at, Chernobyl, or individuals that had exposure to high dose radiation as a child, usually for treatment of another primary cancer. Interestingly, although we don't know all the details, immune factors do seem to play a role in risk of glioma. And it's a little bit opposite to what you might think. So many studies, and many of them quite large, have found that a history of allergy, eczema, whatever it might be, a history of allergy is associated with a lower risk of glioma. And that is very uniformly found across a wide range of studies. And it's something that those of you that follow research on glioma know that trying to interpret and manage portions of the immune system is one place of research interest in trying to manage glioma. Another thing that we have become interested in, and a wide range of folks were interested in the past, but it was very difficult to measure, is whether there is any role for occupation, at least long-term, and/or environmental exposure and risk of glioma. In the past, that was very difficult to measure, in part because we measured things indirectly. So if we thought the water was a problem, you were not measuring the person directly, but you were measuring water sources in their community. If you thought pollution or air was a problem, you weren't measuring the person directly, but you were measuring sources, pollution sources, or Environmental Protection Agency measurements in the community or near highways or cities. We're now better able to measure some of those things directly in the tumor and in the blood or saliva of patients. So in general, although we've looked, including our group and a number of other groups, for a lot of environmental and genetic risk factors, we have not found a lot. We do know that genetic risk factors do explain a proportion of inherited risk. We think it's about 25% of glioma risk. And a lot of people will ask, well, if I have a family member affected with a brain tumor, should I be screened, or patients themselves concerned for their children or siblings or family members? So we estimate that hereditary cancer syndromes only account for about 1% of adult glioma cases. You've probably heard of some of these syndromes, which are relatively rare. People that have these syndromes do have a high risk of brain and other tumors, but typically these families are readily identifiable by the number and type of cancers or syndromes that we see within the family. So many times people will ask, as I mentioned, should I or a family member be screened because a family member has a glioma? Overall, individuals that have a family member with glioma are at twice the risk of the general population. However, there is no recommendation for screening for family members of a person with glioma, in part because although the risk is 2 times the general population, the baseline risk is very low. And this is just a little bit of a picture from a nice article to understand inherited genetic risk, which again is freely available to anyone who might want to look at it. explaining why it is done for family members of breast cancer patients related to the breast cancer gene BRCA1 or BRCA, but not done for glioma. And that is in part just because the baseline risk of breast cancer is so much higher than the baseline risk for glioma. So I mentioned a little bit about the environment. There are now means of looking at tumors from whatever organ they might come from, the brain, the breast, the lung, and identifying exposure to something in the environment and being able to tie, at least with a high level of certainty, an association with that exposure. So, for example, when we look at lung cancer tumors, we can see evidence based on the genetic or mutational changes associated with smoking. For skin cancer, we can see exposure to UV. We can also see exposure, and we've seen that in glioma, associated with treatment. So one of the issues is some of the treatments for glioma, although they may be helpful, they may also cause a problem, including what's called hypermutation, or large number of changes in the tumor that may or may not be helpful when a recurrence or additional treatment is needed. So we have a study now. What we did, we looked at about 1,000 glioma tumors, and we looked for evidence of these exposures across them. We found, not in everyone, but in a small group, evidence of exposure to something called haloalkane. And so that is basically a common chemical in flame retardants. And those flame retardants are not only seen by individuals such as firefighters who are exposed to high levels over a long period of times, but at least in the past, these are materials that coat clothing, furniture, rugs, lots of different things that are in the home. So what we are now undertaking in collaboration with researchers at the University of California, San Francisco, including Margaret Wrench, who led the adult glioma study, we're looking at a small series of firefighters, and we have their tumors, we have their blood, to look and see if we can find evidence of exposure in their blood and their tumors to these chemicals, because obviously that would be very helpful in developing preventative measures if we know these things are likely to be associated with brain tumors. So we are also interested in increasing our sample size. So if you do know of any individuals that have been involved in firefighting and have a glioma, we would love to join up with them. So what is happening in low-grade glioma research? In the past, quite frankly, there has not been much, but we are very excited to talk a little bit about our new project. And Liz Salmi, who will follow me, is also going to talk from the patient perspective how one might advance low-grade glioma research. So the Cancer Moonshot, which is funding from the government, has been reinvigorated. And we, as well as a number of other researchers, have received funds to study cancer, with our project being focused in low-grade glioma. So the network that is funded has a long name, Participant Engagement and Cancer Genome Sequencing, or PECGS network. It is funding 5 research centers and a coordinating center. The cancers and tumors that are being investigated are variable. Some focus on children, some focus on adults. We are the only group to be focusing on low-grade glioma. And as far as I know, we are the largest study of low-grade glioma anywhere. And I don't need to tell you, but basically our focus is on low-grade because there really has been no large-scale effort to study this group. We know about 30,000 individuals, and that's just in the United States alone, carry this diagnosis. And really not too much has changed in terms of treatment, outcome over the past 40 years. So we think it's time to change that and move things forward. So we've created a couple of things. One thing is the low-grade glioma registry, which I know many of you here today have participated in, and we are very thankful for you for doing that. And so why did we go to a registry? Well, we've run lots of other studies, and typically what we've done is we go to either our hospital or our state-based cancer registry For cancers that are more prevalent, you can simply go to your state registry and you will obtain enough individuals who have the cancer of interest. For example, in the past, I've studied breast cancer within the state of Connecticut, which actually has the oldest tumor registry in the country. And using only that state registry, we had enough participants to do a large study. Why do you need a large study? Because you need enough statistical power to get the correct answer. What we've done with the registry is basically move beyond that to broaden our reach. We know we need to go outside these registries. And the other issue is when you work with registries, you typically have to ask a healthcare provider for permission to approach a patient or participant. So many times participants were never included in studies and never knew they even had the option to be included in studies. And so our effort now is basically to just quote unquote, go to the people, go directly to the participants and ask them to decide if they would like to join us. So we're basically using social media. We do still use, including the institution that I'm affiliated with, Brigham and Women's Hospital in Boston, but also to use social media to work with wonderful patient organizations like the ABTA to get the word out there and to help us enroll participants. So the International Low-Grade Glioma Registry, and we are allowing people to join us from wherever in the world they may be. The focus is adults 21 to 79. We do have to make sure that we confirm pathology. We're trying broadly to include categories that, although maybe don't exist now, did exist over the past 10 or 20 years, because we want to make sure we include everyone. There's no restrictions: sex, gender, race, date of diagnosis, place of residence. Basically, you can come from anywhere that you find out about us: the web, social media, hospital-based, word of mouth, friend, etc. And I do have to give a shout out. Where it all began was at the 2016 National Patient and Family Conference. The ABTA gave us space and facilities to start registration. And I don't know if any of our initial members are on the webinar today, but we thank you very much. You can see this is where it all began and has now got to the point where we have over 600 individuals in the registry. So what we've also done is partnered with someone who I think some of you may know, Roel Verhoek, who was at Jackson Laboratory and now happily is moving in the next month or so to join us at Yale University. We know that over time, gliomas may recur and that people have a wide variety, particularly for low grades, a wide variety of treatments. Some people have just observation. Some people just have surgery. Some people have a mix of surgery, chemotherapy, radiotherapy. And so what Roel was interested in doing, he created the Glioma Longitudinal Analysis Consortium, or GLAS for short. And that allows us to look and see what changes happen over time, what treatments worked, what treatments didn't work, what treatments probably caused more problems than helped. His focus, however, and this is in part because higher grade tumors are more common, his focus was initially in high-grade tumors. So what we've done for our new study, which we call OPTIMUM, because as you know, every study has to have a fancy name, our study merges the low-grade glioma registry, which we developed, and GLAS. So the concept now is that we are looking at lower-grade patients over time. As they recur, learning what happens, what changes occur, what benefit or risk is there to doing surgery, chemotherapy, radiation, when should we do it, what should we do, and also learning about what are new treatments that we might be able to look at. And a key portion of this study, as you saw from the title of the network, is patient engagement. So we have 3 goals. As I mentioned, this is a moonshot initiative, and you can go to our website as well as the Moonshot website if you want to learn a little bit more. The first goal, and Liz Salmi, who will speak next, is involved very much in this, is trying to engage participants and patients and other stakeholders, be it family members, physicians, from the lower grade glioma community. We know how important this is, and basically we know we cannot get this done without these individuals. Participant engagement. So this is the section that I run. Our overall goal is to develop a cohort, which is a group of 500 lower grade glioma patients who have had surgical recurrence. The number is selected because we know there's enough variation between or among patients that to get a good picture of what's happening for those who have chemotherapy, for those who have surgery only, we need a certain number to get a feel for what's happening, what changes occurred, what did we do right, what did we do wrong in treating patients so we can move forward. And then AIM 3, which is run by Dr. Verhoek, who's, as I mentioned, at Jax and now going to Yale, is to actually do genetics on germline, which is blood or saliva, and the tumor. And what's unique about our study is we're going to deliver these results to the participants. So these are the questions that are of interest. I've mentioned most of them, but I know some of you have already come up against these questions. What is the benefit of surgery? How does that work? Should we hold off on treating people with chemotherapy? Are we causing more problems with alkylating agents like PCV and Temodar? When you have a recurrence, should you have surgery or can you go straight to being treated? And what role does knowing about these genes tell people? So what do we ask you to do? This is our site. We hope it's not too much. There's an online questionnaire, consent. We will locate your pathology and medical records and ask you to give us a blood or saliva sample. And if you don't have access to the internet, we can do this with you over the telephone. What do we offer? Well, we're hoping that by joining a group of persons with glioma, that we will offer individual and group level information, not just on things that are relevant to you, but what other folks in the community are doing, what's new, what's novel. And we're hoping to advance things, and you'll see later in today's meeting, neurocognitive effect. How can we help people with disability insurance? Are there diet and exercise things that people can do? And then also we'll return the results, both individual and study-wide. So as I mentioned, we have 602 patients in the registry, 87 with recurrence. This is the picture of where everyone is. You can see we're pretty good in some states, but we're not too good in other states. And we hope you will enroll and join us. We also have overseas participants, mostly from English-speaking countries, but as we've been mentioning, we're hoping to change that with the addition of Spanish and hopefully other languages. And we have received funding from NCI to translate our materials. We're working with ABTA to translate both written and virtual materials. We're going to translate the questionnaire, and we're hiring a project director at Yale. Actually, he was the young man who created the introductory slides, and he is bilingual and a certified translator, so he will be helping us to do so. So how do you join? This is our website. If you have any problems, you can contact me, or any questions you want to learn a little bit more before you go anywhere or think about anything, glioma@yale.edu. We are registering people at different hospitals as well, Mass General Brigham, UCSF, Dana-Farber, and we're hoping to add additional sites. And please also follow us on Twitter, Facebook. Our website is at Glioma Registry. And thank you. I will now turn things over to our star of the show. So it has been my great pleasure to work with Ms. Liz Salmi. Many of you probably know her. As her slide says, but the slide does not convey even a tiny proportion of all that she does. In her work hat, she is Communications and Patient Initiatives Director at Open Notes, located at Beth Israel Deaconess Medical Center. But that is only the beginning of things. You probably know, and you might be able to tell from her hair, which is even a different color today. Her past, she was a drummer in a punk rock band. She is a tireless patient advocate. She is a person living with lower grade glioma. We have so enjoyed working with her that we begged her to be a co-investigator on our OPTIMUM grant, which she kindly and probably is now rethinking things, agreed to do. She is also, which most of us in the research community cannot say, a first author at New England Journal of Medicine, and I highly recommend that you view her recent perspective on surgery for glioma called Deciding on My Dimples in the New England Journal of Medicine. So it is my great honor to present our co-investigator at OPTIMUM, Liz Salmi.
So much hype. Thank you so much. I want to make sure people can hear me. Yes, Dr. Claus, sounds good. Okay. And also I think I might have control of the slides, but if I don't, I'm clicking start to get the remote control. Give me a moment. Okay.
There you go.
Hey, there we go. No disclosures. I have no stocks. I am on the board of directors for National Brain Tumor Society, and I'm supported by grant funds. And to let you guys know, I'm Liz Salmi. I'm the Patient Initiatives Director at Open Notes at Beth Israel Deaconess Medical Center. And my work in research is really focused on engaging diverse stakeholders and the design and conduct of research. But it's important to know that my whole work and world around research comes from a patient perspective. I'm a person diagnosed with a low-grade glioma and astrocytoma in 2008. And then I had a recurrence in 2009 and did the whole surgery aspect of things. And that inspired me to get involved in research and really think about what can patients do after our lived experiences and really level up and learn more and how we can participate in programs like the low-grade glioma registry. Actually, the low-grade glioma registry inspired me to become a researcher. But as we know about low-grade gliomas, astrocytomas, oligodendrogliomas, they do recur. And that happened to me. Actually, just in a year and a half ago, my glioma recurred. Thankfully, it's still grade 2 astrocytoma. And then it recurred again less than a year ago, and it's still grade 2 astrocytoma. So over the last, really just my experience, and even becoming a researcher, I'm not immune through this experience. And so over the last 15 years, I've really had the luxury of time to think about why these tumors recur, and I've had questions, and it's made me think and wonder, how might brain tumor patients like us influence research priorities in partnership with those who are studying our diseases? That's what really lights me up. And the idea of patients partnering with researchers is not science fiction. It's not a pipe dream, because it's actually a reality. Patient advocates with other conditions besides brain tumors have been leading the way. They're evaluating research proposals, helping establish research agendas, and actually some patient advocates have received funding to lead their own research, which is always super inspiring to me. So really what I'm saying is there is this growing space where patients are partnering in the co-design of research from the beginning of the research process all the way through the end to disseminate that. And this concept of partnership is called stakeholder engagement. So if what I'm telling you sounds too good to be true, it's already happening in other health conditions. We see this in type 1 diabetes. We're seeing this in breast cancer. We're now seeing this in long COVID. So I'm excited to share that we're working on this approach in low-grade glioma, specifically with the low-grade glioma registry and the OPTIMUM studies with Dr. Claus, which just told you about. These studies are run by researchers at Yale University, University of Colorado, Jackson Laboratory, Mass General Brigham, and where I'm at Beth Israel Deaconess Medical Center. But even though we're running the study, it means people from anywhere receiving treatment anywhere can actually participate in the US and in some cases overseas. So these two studies, there are two national studies of people with newly diagnosed and recurrent low-grade glioma, newly diagnosed, and I'm recurrent. And you can participate if you have an oligodendroglioma or an astrocytoma, and you're between the ages of 21 and 79. So if you're here, you probably qualify for one of these studies. So we'd love you to join us. And we really have a big vision in this work. It's to involve participants like us along the way in the study to make it easy to participate. I'm all about making it easy and really to share what we learned back with you. So you're not in the dark going, what's happening with the study? We want to share with you our progress. And this registry has a number of goals. We want to know why some people develop low-grade glioma and some don't. I'm really curious about that. We're looking at variations in inherited tumor genomes, like doing the science. And then, as Dr. Claus shared earlier, this registry data will help us understand how lifestyle factors such as diet and activity, occupation might be related to glioma risks and outcomes. And from my perspective, what makes these studies really unique is how we're involving you, the participants, along the way. Rather than research being something that's done to you, we're working on making this research something that's designed with you. And so, for example, we have the Low-Grade Glioma Research Advisory Council. We've existed for about a year now. I'm mainly involved in that work. We've got 19 people with low-grade glioma directing and advising on what we want to see involved in the work. And then a number of diverse group of individuals. So we're working with the LGG community in this study for over a year now. And while the low-grade glioma registry has been around for more than a decade, we're really ramping up the participant experience. So folks like you, be part of it. We want you to get involved. The data reflected on the slide is actually outdated. It says 400 individuals enrolled in the registry, but actually, as Dr. Claus announced today, there's more than 600 individuals enrolled in the registry, and I think more than 60 folks who are involved in phase 2 about the recurrent part of the registry, and we want you to join us. So we have a website, it's gliomaregistry.org. That's the main space to go and learn everything about the study, download consent forms, share it with your doctor, or just, go there and you can start signing up yourself. We just really I want to make it an easy experience for those of you who are going to be participating. And really, here's the call to action for all of you, is that in order to make advances in low-grade glioma research, we need to grow a larger, more representative group of participants in order to really study this stuff over time. So if you're not in the registry, we need your voice and your participation. This 600 participants is an incredible foundation, but we're looking for everyone to be signed up and to get counted. And there's a number of ways to get involved. I already mentioned the website. It's gliomaregistry.org. Go there. We're looking for newly diagnosed, as well as those who are like me, or recurrent over time. You could send this information, gliomaregistry.org, to your doctor to say, hey, help me get enrolled. What do you think? And then you can also share the registry information with the patient community. So once you're signed up, tell your friends. I mean, I know as a patient myself, I share information on Twitter and Facebook, and I talk about the registry all the time. So that's how I try to get the word out myself. Finally, I just want to recognize the time, so I'll wrap things up. But again, I'm Liz. You can reach me at my email address here. I'm also on Twitter as @TheLizArmy. All right, I'll wrap things up, and I guess we're going to Q&A. Thank you so much.
Yes, thank you, Dr. Claus and Liz, for that wonderful presentation. As a reminder, if you have a question that you'd like to ask, please type and submit it using the Q&A button on the bottom of your Zoom panel, and we will answer questions as time allows. All right, so one of the first questions we have here is asking, Dr. Claus, I'm going to direct this one to you. You had talked about some environmental factors that may impact diagnosis of low-grade glioma. Is there any study or any link found between the tragic events of 9/11 and a diagnosis of low-grade glioma?
There is definitely, as I'm sure many individuals know, a study following up the survivors and individuals in the direct area of the 9/11 events. And higher reports of cancers have been noted, but not yet in lower grade glioma. It's more cancers that are, more highly frequent, lymphomas, leukemias, and that sort of thing. But I know they will continue to follow these individuals for many years, so we'll have to wait and see.
Thank you so much. And Liz, you shared some wonderful ways that our patient population online today can get involved in research and participate. Can you tell us a little bit more about how extensive the participation is? So what does it entail for a patient to participate in the LGG registry or the OPTIMUM study? How much exactly are they expected to do? What does that look like?
Yeah, we're trying to make the participant experience really easy. I mean, if there's this high barrier of entry, it's hard, and we want people to participate. So if you go to gliomaregistry.org, you learn about some background information about the study. People should be informed, and really, each of us can go straight to the website and click a button in the upper right corner that's about join the registry, and then that brings you to a secure platform hosted by Yale that allows you to start and basically filling out a really long, comprehensive survey designed by Dr. Claus and her team that asks for, what's your name, this, that, and the other thing, and some treatment history, and leads people along the process in order to finalize the whole enrollment process. From there, once your information has been, entered into the system, you should expect a follow-up messaging from Dr. Claus and her team at Yale, asking questions. They will be helping people through the process of collecting medical records. And so it does take some time and leaning in for us. But at that point, you're in the full study, and you will receive updates and messages about how things are going with it along the way.
And I think I'm just seeing a couple of questions, and Liz had mentioned this, but I wanted to be clear that you can join our studies whether you have had recurrence or not. So the low-grade glioma registry is for any adult with lower-grade glioma. And then that subset of individuals that have recurrence would be eligible for OPTIMUM. And I saw someone from Pennsylvania wants to join and represent, so please do.
And just to say, before I was a researcher working on many projects, including now a partner with Dr. Claus, and I was enrolling for the first time, I did the process of filling out the survey and did that. And then having the more recent recurrences, it gave me this weird opportunity to enroll again in the recurrent part of the study. So I've had a chance to see both experiences. So really, you start at the initial enrollment period, and then if you have a recurrence, come back to us, because we want to make sure you're continuing to be counted.
Thank you both so much for that insight. Dr. Claus, please correct me if I'm wrong, but I believe you had shared that these studies are available internationally as well for patients outside of the US. Can you share what that looks like for someone who is international wanting to register? Are they having to mail in these materials? Can they complete it online? And if they do have to mail in materials, do they have any assistance with that?
So all of the information that we gather, for the most part, can be collected digitally online. So consent, the questionnaire is all done online. We've been having great success with using saliva kits, and now we're also going to trial blood spots, because we know probably getting whole blood samples in a tube is not going to be a reasonable thing to do overseas. So we do send the kit to individuals. What we try to do is, if you identify where the surgery was and you give us written consent, we then try to approach the hospital to obtain the materials and pay, the pathology department for whatever materials are needed. Sometimes we do overseas need a little help because they're not used to having American or overseas individuals ask for materials. Interestingly, we do have I see there's someone from Turkey. We have a large number of collaborators from Turkey. Neurosurgery in Turkey is incredibly strong. And I do have a number of collaborators over there. So I think we could certainly also work with them to help facilitate things. You may already even be receiving care from those individuals who are very happy to work with us. So.
Thank you so much. Dr. Claus, I'm going to direct this one to you as well. We have a question here asking, if someone is already part of the registry, how can they update their information if they've had progression or are starting new treatment?
So the easiest way probably is to contact us at glioma@yale.edu, and we're happy to update everything. And we do appreciate it. Many times people do shoot us an email or leave us a voicemail to say, hey, either a family member was diagnosed with something different or our treatment has been updated. And we really appreciate that. It helps us very much. And also it might make you eligible for the OPTIMUM study if you weren't before.
That's how I got in.
Great. Thank you so much. Liz, we have a question here for you asking, if you don't mind, can you share a little bit about what treatments you've undergone?
Yeah, for sure. When I was first diagnosed, 2008, we had like different protocols in the neuro-oncology world about what was the best course of treatment for newly diagnosed, or I was 29 at the time. And then things have changed in the ensuing 15 years. And new World Health Organization guidelines have come out that say newly diagnosed folks should go through X, Y, and Z path. So my experience, just to say, might not be what your experience is in the last few years or even today. So I'll put that out there as my disclaimer. I've been, 15 years living with grade 2 astrocytoma, and it stayed the same. So my treatment path all that time, age 29, it was a watch and wait period. So did nothing. And then 6 months later, tumor recurs. And then I was, at the time, the guidelines of radiation versus chemotherapy, what should someone do at that age was kind of up in the air. So my path was to do temozolomide. I did that for 24 months or 2 years and then stopped. And then there was a long drought of no changes for 12 years. And then I learned lots of stuff and started working with Dr. Claus. And then I had a recurrence in 2021. I have a problem talking about numbers, 2021. And that's where I thought, oh my gosh, things must be progressing and becoming more malignant. But as a result of that surgery, we learned I was still grade 2 astrocytoma, did nothing. And then 6 months later, the tumor recurred. And at that point in time, finally, I was a candidate for radiation. So I did radiation 24 months. And then I followed it up by temozolomide for I did radiation for 6 weeks, did radiation for 6 weeks, and then followed that up by temozolomide for 6 months. So that's where we're at. Actually finished temozolomide a month ago. So I'm back in the game as a person just living with the brain tumor. Long story, but you guys know what I'm talking about.
Thank you so much for sharing that, Liz. Dr. Claus, we have another question for you. This participant was originally diagnosed with a low-grade glioma, was treated, and then was diagnosed with a different tumor type. How often does this happen? Is it common for someone who's had a low-grade glioma to then in the future receive a different tumor diagnosis?
It changes over time, and part of this is because what we know about gliomas has changed over time, so we're better able to update what the true diagnosis is. But what we're doing in our study is we will re-look at everyone's initial and secondary or subsequent tumors, and using the updated guidelines, go through and see what's happening.
Thank you so much for answering that. Another question we have here is asking about the new classification of brain tumors. For a patient who is diagnosed prior to these changes, will they receive an update from their healthcare team about how their diagnosis classification may change, or is that something that they should be talking to their healthcare team about to better understand?
So it's going to be variable, where you receive your care. In some institutions, this would be done automatically. I would say in many institutions, it will not be done automatically. And so you would need to request from your, probably neuro-oncology would be the best person to request it. And Liz, I don't know if you have any experience with, I know that for some of your cases, people always now go back, retrieve, if possible, old samples to update them to kind of learn, well, what really did the person have 10 years ago?
I was, I don't want to make this a super long story, but I think this falls in the realm of patient advocacy. To some extent, you see things like gliomaregistry.org. It seems like it's an easy process of, yes, I'm just going to send my samples and they'll magically appear. But one of my motivators for making the registry, as well as the OPTIMUM part of the study, easy for our participants is I went through that experience of going to my old doctor at a different health system and saying, can I get these tissue samples so I can get them off to the registry? And that whole experience, my doctor at the time said, why do you want these tissues? I'm like, so I can participate in research, and I could, send it off to this person at Yale, and she'll find the answers to things. And my neuro-oncologist, who didn't know anything about the registry, really was like, well, what if we find out something you don't want to know? And I was like, but I want to know things. So they made me wait in order to get that, do that whole medical records request. And so I did wait the 6 months to get the medical records. And finally I said, I want this information. And so they then allowed me to get this information passed over to the study and the investigators. And really that experience of waiting and asking and making it hard is what inspired me to make sure our work with the low-grade glioma registry, and then the parallel cousin study of OPTIMUM, I want to make it an easier experience for all of us. We're still figuring out what that process looks like and trying to make sure it's a smooth ride, because we're still in the early first years of testing out the process, but I'm super excited that we're going to achieve that goal.
Thank you so much. We have a question here from one of the caregivers. They're asking, how can they get involved in research? Is there any role that they can play in the studies that both of you have discussed today?
I think we would both say, absolutely. We need all the help we can get, even just getting the word out there. Or, as the ABTA has this mentor-mentee program, helping others to participate. And Liz, let you say your thoughts as well.
I mean, people living with brain tumors, we have different levels of capacity over time. Right out of surgery, I can't do all of the things. My husband is involved in helping me. So our care partners are absolutely crucial to, as a patient, you could say, hey, I need your help. I'm interested in doing X, Y, and Z project, or so at certain phases in time, we might need help, or we can enroll in things like the registry on our own. So care partners can either help us, guide us through the process. Sometimes we need help reading, writing, language. I'm still struggling with numbers. So those who help us with the regular care are natural partners in enrolling and partnering in research.
Great,
So,
thank
yes.
you so much. So is a follow-up question to that, are a patient and caregiver duo able to join the study together so that the caregiver can step in and share information when the patient is unable to do so?
Absolutely. So even on our consent, we allow for a proxy individual. Yes, so absolutely. And also you can, benefit from us. I am lucky enough to be at Yale within the Yale School of Public Health. So we have hundreds of wonderful students, including some who have, helped put pieces of this together. And, they work with us. They're very willing to help assist people who can't do internet or don't want to do internet. We can do things over the phone. There's all sorts of ways we can make things easy for you. So yes, however you want to come hang with us is how we would love to have you.
Wonderful. Thank you so much. Another question we have here is asking, once a patient has submitted their information, their pathology and whatnot, do they receive a confirmation that this information has been received?
So we do tell them via email that the information has been received. The one thing, and I know, Liz talks about this quite a bit, in terms of us getting the tumor tissues, we've had an overall very good rate of success, but it can take us a long while. So it can take us 6 months to a year to get physically in our hands the tumor tissue at outside institutions. We do after that tell people that we've received it, but that can be a long time. And I know sometimes people, they're used to kind of an internet age. They would like for things to be moving a little bit more quickly, but that is the reality of how long it takes us to do things. And Liz, I don't know if you want to say.
As a patient, a person living with low-grade glioma who's involved in this study, I have been shocked by the speed of research. It is so slow. Like, I had no idea how much time it takes for researchers to do their thing. And that makes sense. There's ethics and science is happening here. So I've learned so much. My dream state for research, which is not going to exist at this time, is we would have something like the Domino's pizza delivery app, where you could say, I've submitted this, my study is samples in process. It's being, your pizza is delivered coming to your house and it'll be there in 15 minutes. Like, I wish something like that exists. It doesn't, we're not there yet. We're looking at months, perhaps years, till the point where we're going to see a result in our hands, but it's happening. Like, that's the whole point of studying this process, especially, for the registry and both the low-grade glioma registry main and also OPTIMUM study, is really looking at that and then engaging with our research advisory council, the patient advocates, on how do we want to receive that information. And that's a component of what we're doing.
Thank you so much, Liz, for sharing that insight. Unfortunately, that is all the time that we have for questions. If you do have important questions, please continue to submit them, and our speakers will try to address them as they can in the Q&A box. I'm going to go ahead and turn it back to Emily, and we'll move forward with the rest of the program.
Yes, thank you so much, Dr. Claus and Liz, for joining with us today and sharing the importance of patient engagement in research to advance treatment. That was such a great question, and thank you to all of you for your wonderful questions. We will resume the program in about 5 minutes for our next session titled Novel Treatments and Clinical Trials. We'll see you back soon.